is operon. Operon is a functioning unit of DNA containing cluster of genes under the control of a single promoter. Lac operon concept explains the metabolism of lactose in prokaryotes. It consists of various genes help in lactose metabolism by producing different enzymes. Before we move into the concept, let's understand the terminology. The P indicates promoter, the O indicates operator, the I represent regulatory gene which codes for repressor. The LAC-Z, LAC-Y, and LAC-A are structural genes code for some enzymes. The gene I is called regulatory gene which codes of the repressor of the Lac operon. Repressor is a small protein found by the regulatory gene which binds to the operator and block the passage of RNA polymers towards structural genes. Once the repressor binds to the operator becomes switched off. In the presence of inducer, nothing but lactose which attaches to the repressor and changes the shape of operator binding site, then the operator becomes free. As soon as the operator gene becomes free, RNA polymerase is recognized by the promoter Z, then RNA polymerase passes over the operator gene and then reaches the area of structural genes, then operon is switched on. Structural genes take part in synthesis of polypeptides, protein, enzymes through the formation of specific mRNA. This locked mRNA is known to polycistronic, which produces all three enzymes called beta-galactosidase, permis, and transacetylase. Beta-galactosidase is primarily responsible for the hydrolysis of the disaccharide. Permis increases the permeability of the cell to beta-galactosidase. Transacetylase catalyzes the transfer of an acetyl group from one molecule to another. The trp operon consists of regulatory and structural genes. The promoters or operator region of the trp operon regulates the level of transcription. There are 5 structural genes, labeled trp E to trp A, are located downstream of the promoter region. The structural genes are transcribed as one of the polymeric mRNA. The polymeric or polypeptides translated from the mRNA are responsible for tryptophan biosynthesis. Between the promoter and operator region and the structural gene is the leader region called trp-L. This region is transcribed into an mRNA called the leader mRNA. The leader mRNA contains a short open reading frame that is translated into a leader peptide. The leader mRNA contains the attenuator consisting 4 separate regions that can fold and form 3 different stem loop secondary structures by complementary base pairing. Pairing of regions 1 and 2 result in a transcription pause signal. Pairing of regions 2 and 3 result in anti-termination signal that allows transcription to continue. Pairing of regions 3 and 4 results in a termination signal which stops further transcription. Because there is no nuclear membrane in bacteria, transcription and translation are coupled. Translation of the trip mRNA can proceed while RNA polymerase is still transcribing the attenuator mRNA. The position of the ribosome along the leader mRNA plays an important role in the regulation of transcription. After RNA polymerase has transcribed region 2, region 1 and 2 can pair to form a stem loop structure. This structure causes RNA polymerase to pause and allows the ribosome to catch up to it. Subsequent transcription is tightly coupled with translation. Now, regulation by attenuation can occur. If the cell is starved for tryptophan, then the amount of tRNA drops dramatically. In the leader mRNA, there are two trip codons in tandems. When the ribosomes reaches these codons, it stalls because the specified amino acid is in short supply, and the leader peptide cannot be completed. Since the ribosome cover region 1 of the attenuator, region 2 will pair with region 3. This pairing is called anti-termination signal, thereby allowing RNA polymerase to proceed, and translation follows to synthesize enzymes necessary to produce more tryptophan. If the cell is not starved for tryptophan, there is enough tRNA for translation. When the ribosome reaches two trip codons, two trips are added to the leader peptide, this one. It will add, assume that there is a addition of this drawing. The ribosome then continues to stop codon at the end of region 1 of the leader peptide. This prevents region with region 4, which is a termination signal for RNA polymerase. So the ribosome and this one will be faded. And that is the process of trp operon.